who I introduced previously, Dr. Wei-Ching Zong. He received his PhD from the University of Medicine and Dentistry in New Jersey, which, as you know, is now part of Rutgers University and specifically RBHS. Wei-Ching did his postdoctoral work at the University of Pennsylvania. The title of his presentation today is The Nitrogen Side of Cancer Metabolism. Wei-Ching, the floor is yours. Thank you.
Thank you, Stephen. So can you hear me, see me, all right?
You are good to go.
Very good. So thank you, everyone. So we have heard quite some about metabolism, glycolysis, mitochondria, so about carbohydrate, right, carbon. And so now today I'm going to share our work in the past few years on another important element, nitrogen, in terms of cancer metabolism. So let's see. Forward. Don't seem to. Am I stuck here? So somehow. Oh, okay, here. So nitrogen as a essential element, right? Even though there's almost 80% of nitrogen in the air, we as mammals we cannot directly fix it or assimilate it. So I have to take nitrogen from our food. So the fate of the nitrogen containing food, especially protein, gets into our digestive system, especially gets digested in the gut. And then the excessive amount of protein content in the food will be actually converted in the gut, in the small intestine, by microbiome to the nitrogen waste in the form of the ammonia. And then what happens with ammonia is that ammonia needs to be converted into the non-toxic molecules. So firstly, after ammonia enters the liver through the so-called portal vein, ammonia is converted to urea for excretion, and then the blood still contains the residual amount of ammonia, keeps travel through the lining of the hepatocytes, and then eventually goes out of the liver. So right before it goes out of the liver through this hepatic vein, so near the perivenous area, the residual amount of ammonia is converted to glutamine through this glutamine synthesis, and then that will help to detoxify the residual amount of ammonia. So basically, these two processes, urea cycle and the glutamine synthesis, play an important role in the liver to detoxify the toxic ammonia in our circulatory system. So if there's any defects from, say, liver injury, then ammonia gets into the brain and causes major problem in the brain that's called hepatic encephalopathy. So just some historical blurb about this ammonia detoxifying mechanisms. Actually, it was discovered by this person, Hans Krebs. So this name may sound very familiar to you because he is the one who actually illustrated the TCA cycle, right? The very famous mitochondrial respiration cycle. But his other major discoveries are he found the urea cycle, and then he discovered the glutamine metabolism, both enzymes, glutamine synthetase and this glutaminase that breaks glutamine into glutamate. And also he discovered the amino acid oxidase, also we call them deaminase or transaminases. So coming to cancer metabolism, right? As we have already heard, especially from Josh's first talk, right? There's abnormal cancer carbon metabolism in the cancer cells. So we all know to this audience, right? There's an increased glucose consumption through this Warburg effect, and also this TCA cycle, right? Identified by Krebs when he actually was a research assistant in Warburg's lab. And then in addition to that, in recent years, this glutamine has been talked about or has been discovered as another major alternative of the carbon source to fuel the TCA cycle. So what people have demonstrated is that some oncogenes can upregulate the uptake of glutamine and then its conversion into glutamate through this glutaminase, and then glutamate can be can then lose another amine group, and then it can be converted to alpha ketoglutarate to enter the TCA cycle. So this glutamine to glutamate to alpha Kg reaction has been described as a major carbon usage pathway for cancer cells when glucose supply may be limited. So, and then there's quite some effort in developing inhibitors to inhibit this enzyme glutaminase, and there has been in several ongoing clinical trials. So with that, a few years back, my lab actually made some quite intriguing observation that in several cancer types, we found that instead of this glutamate alpha Kg upregulation, when we look at say MYC activation in hematopoietic cancers and also in breast cancers, we found actually there's an upregulation of the reverse reaction, meaning the instead of showing the induction of the glutamine to glutamate to alpha Kg, we saw the reverse reaction is induced by this MYC oncogene, and then we further figure out this is through MYC-mediated activation of this DNA demethylase, and then that caused the demethylation of this promoter of the enzyme that's called glutamine synthetase. So this was very surprising to us, and then we were trying to find out, okay, what's going on here, right? We've made a essentially an opposite observation. So we look at what's going on with this enzyme. It's also called GLUL, glutamate ammonia ligase. It's another name is glutamine synthetase. So I'm going to use these two names in the following slides. So we look at several tumor models and then look at the patient data. We found actually that this GLUL or GS expression is highly induced in this PDAC, the pancreatic ductal adenocarcinoma, as described already by Josh earlier. So in the several versions of different PDAC models, we see the induction of this GS in the tumor area. Okay, so also this correlates very well with what we have found in the patient in the clinical samples. So obviously the experiment, right, to do is to delete it in the pancreatic cells. So we use this PDAC model, bred it to this flox GLUL mouse. So we can see this deletion of GLUL in the pancreas. And then surprisingly, or not so surprisingly, that by deleting this GLUL, we see a much better outcome so much slower tumor development and the better animal survival. So right, so we did a bunch of mechanistic study, and then to explain this, right, what happens is that even though glutamine can be converted to glutamate by losing this terminal amide group, then this can be further converted to alpha Kg to fuel the TCA cycle. But what we believe is in a situation where the blood supply of glutamine is limited, for example, in the highly fibrotic pancreatic cancer, right? This glutamine supply from the circulatory system actually is highly limited. And then we cannot forget that this terminal amide group actually is the essential donor for making this nitrogen-containing molecules, purines, pyrimidines, NAD asparagine, hexosamine. So this is the nitrogen donor in our body, and also for the cancer cells, in order for them to survive and then to proliferate and then to do DNA replication and DNA repair, all that. So around the time when we show our work, there are several studies showing that indeed this reverse glutamine anabolic process reaction plays a very important role in different cancer types under different situations. So to summarize this part of the work, right, we show that instead of this glutamine catabolism reaction, in certain cancers, for example, such as the PDAC, this glutamine anabolism reaction plays an important role by making the cancer cells being able to utilize this nitrogen group to make nitrogen anabolism to support cancer cell survival and the growth. So now coming back to the liver. So here is the liver, as I already alluded to earlier. This is the portal vein. This is where the blood coming into the liver from the small intestine. So near the portal vein, and then between the portal vein and the hepatic vein, where is the blood leaves the liver, there's a high expression of urea cycle enzymes. So here using RH1, as you heard from Eileen's talk, this RH1 is one of the 5 urea cycle enzymes. So it's highly expressed near the portal vein and then in the middle between the portal and the hepatic vein. And then along the blood moving from the portal vein to hepatic vein, and then this GS expression is high near the hepatic vein. So looking at the human cancer, right, the GS or GLUL is specifically highly induced in the liver cancer. So then we figure, oh, this indicates that this GS may also play a tumor promoting role in the liver in hepatocellular carcinoma. So we were saying, okay, let's knock it out in the liver to see what happens. So then we use this, again, this albumin mediated GS deletion, specific deletion in the liver. And then we use the system, the tail vein, the hydrodynamic injection of 2 oncogenes, c-MET and this beta-catenin. So beta-catenin is a highly prevalent oncogenic event in liver cancer. So this tail vein injection will help sending these 2 oncogenes into the liver, and then as you can see, GS expression is already a very well-known substrate target for beta-catenin activation. So now, in addition to this peri-venous area where GS expression is high, now oncogene expression can induce the GS expression in the sporadic hepatocytes in the liver, and obviously this is not happening in the knockout. So let's see what happens with this oncogene-induced liver cancer development. So to our surprise, now unlike what's going on in the pancreas, now knocking out GS in the liver leads to increased tumor development, right? With increased PCNA and then with much increased liver to body weight ratio. So this is an indication of tumor burden in the liver, and then also much decreased survival. And then we were surprised, and then we look at several other models. This CMET with catenin, so the model I already showed you. And also there's a YAP catenin model, and then we have two carcinogen models, so diethylnitrosamine plus phenobarbital, and then plus high-fat diet. So in all four models, we see increased tumor burden, tumor development in our GS knockout. So one common feature here is that, so these oncogenic stress or carcinogenic stress, they can all induce GS expression. So what's going on here is that when we delete, ablate GS, this promote tumor development. So what's going on? And then one important, right, the cell growth signaling pathway apparently is the mTOR activation. So we look at mTOR through different ways, and here showing one evidence is that in our knockout tumor, indeed, there's a strong activation of mTOR. And then when we use rapamycin to treat the animals, now the liver is much significantly inhibited, almost to the level of the wild type liver. So mTOR activation plays a big role here in this knockout, GS knockout HCC. So then this is kind of contradictory to people's belief, right? Because GS is responsible for making glutamine, and then glutamine is well known to induce mTOR. But how come when we knock it out, we see higher mTOR? So then we look at these liver tissues, our mouse liver tissues, more carefully. So what's going on here is that, so with this beta-catenin activation, right, we see the beta-catenin can induce GS expression. But then almost like superimpose, we can see the downregulation of urea cycle enzymes. So here, this slide shows the CPS1, but then when we look at the several other, including ARG1, ASS1, so all the urea cycle enzymes are suppressed by this beta-catenin. And then we look at the animal plasma glucose, plasma glucagon, there's no difference. One major difference we can see here is that the plasma ammonia is increased, is induced by oncogenic activation, and this is further exacerbated by this loss of our GS. And then when we look at the interstitial, tumor interstitial fluid, right, this ammonia level is even higher. So this leads us to think the oncogene-induced urea cycle suppression may also play a role here in terms of disturbing the ammonia homeostasis. So looking at the literature, we know, right, the liver is the organ that has high urea cycle activity. So the expression of the urea cycle enzymes is particularly high in the liver. And then in the liver cancer, the urea cycle enzymes are actually mostly all downregulated. This is in the TCGA data. And then the suppressed urea cycle enzymes actually correlate with a poor prognosis. So what we believe here is that this oncogenic activation, especially beta-catenin activation, will lead to the failure of urea cycle. That will increase the ammonia waste burden in the liver. Okay, so this will force the tumor cells through some mechanism to upregulate this glutamine synthesis pathway to cope with this increased ammonia stress. So then in this system, right, now if we ablate this glutamine synthesis pathway by deleting GLU or GS, now we get an accumulation of ammonia and also the accumulation of the substrate for glutamine synthesis, which is glutamate. And then the alpha carbon, the alpha amine group of glutamate is the essential nitrogen donor for making the other non-essential amino acids in the mammalian cells. So these several non-essential amino acids, including alanine, aspartate, tyrosine, serine, so they may be induced through this accumulation of glutamate. Then we believe this can stimulate mTOR activation, mTORC1. So using the stable isotope tracing, as you have already heard, we track down where this ammonia waste may go, right, in our liver. So basically, this ammonia can be assimilated into glutamate first, and then to glutamine. So when we look at our oncogene-induced or treated wild type, so wild type is Cre minus, knockout is Cre plus. So we see, right, Cre plus is the GS knockout animal. So not surprisingly, in the liver, the glutamine synthesis is much reduced. And then reciprocally, glutamate synthesis is much increased, and there's a huge accumulation of glutamate. And then there's also a robust increase of alanine and some increase of aspartate and tyrosine synthesis in the liver. And then there's a very little effect on other several other non-essential amino acids, and then there's absolutely no effect on the essential amino acids. And then we are all familiar with essential amino acids, especially the branch chain amino acids activating mTOR. So we look at the using cell culture, right, to find out actually the non-essential amino acids in the presence of essential amino acids can also strongly stimulate mTOR activation in different cell systems. So this helps us to believe, right, this glutamate and glutamate-derived non-essential amino acids may play an important role in activating mTOR. So we here we silenced one transaminase that converts glutamate to alanine, and then we can largely suppress the tumor burden and then also the mTOR activation. And then also using the pharmacological inhibitor of this glutamate alanine transaminase, we almost see identical effects in terms of suppressing tumor. So, so what we believe what's happening here in the liver, then unlike in the pancreas, is that so here in the liver, due to the hyperactivation of beta-catenin, so this will lead to the defective urea cycle, and then this will lead to the accumulation of ammonia waste. And then at the tumor initiation or at the early stage, right, the hepatocyte try to jack up the expression of this glutamine synthesis pathway, try to alleviate this ammonia stress. So if through some mechanism, right, GS is suppressed, and then there will be a accumulation of this glutamate. So this ammonia accumulation, glutamate accumulation will lead to the NEAA increase, and then that can activate mTORC1 to stimulate HCC. So indeed, when we look at the clinical samples, right, we know that looking at this several comparing the tumor tissue versus adjacent normal tissue, we know that although GS is highly suppressed in some cases, showing here in green, there are also many cases actually the GS expression is down. And then importantly, this high GS expression actually correlates with decreased mTORC1 activation, and then the GS high patients actually, especially in the beta catenin mutation mutant. So the gene is CTNB1. In this mutation patients, actually these patients do better if their GS expression is high. So right, this is our new kind of understanding of how GS or the ammonia homeostasis may play a role in different organs, different tissues under different scenarios. So currently we are trying to study how the dysregulation of nitrogen homeostasis, especially the ammonia waste handling, may be tumor promoting, may contribute to tumor initiation and tumor growth with certain mechanisms, how this urea cycle enzymes and the GS expression are regulated at the transcription level. And also from the translational point of view, we believe that lowering the nitrogen waste load may be a good way to consider for liver cancer treatment and the prevention. So with that regard, we can think about maybe the low protein diet, or to manipulate the ammonia production in the microbiome in the gastrointestinal system may help with this. So we have some preliminary data showing actually the low protein diet indeed can enhance the tumor-bearing animal survival. So we don't have yet immunology data in this, but as you can imagine, right, there's certainly a connection with the immune response, especially from the small intestine, when we look at the dietary and the microbiome regulation of the nitrogen homeostasis. So that's what we're looking into next. So with that, I will, here we have a lot of collaborators over the years in our work in the mechanistic basis, and also the pancreatic and the liver cancers. And then the people who did the work in my lab are highlighted in red, and then this the current lab, actually the lab a few months ago. So with that, I will stop, and then I'll be happy to take any